So welcome everyone to this month's GDC webinar. Today I'd like to talk to you about GDC download and upload strategies. My name is Bill. I am the lead for the GDC user services team, and I'm not putting this in presentation mode right now because this is going to be a demo heavy webinar. So I'm going to be going back and forth between text boxes and the command line and web browsers. So I'm going to leave it like this. So just to give a bit of an overview of what will be talking about today. As usual, I will give an introduction to GDC for anyone who's new or needs a reminder about that. Then I will talk about some GDC download tools and demonstration for using several different methods, as well as several different methods for uploading to the GDC. Afterwards, we will go over some frequently asked questions that I've gathered based on some help desk queries that the GDC has received. So to move forward, I'll just talk about the GDC a little bit. Basically, the GDC is a data repository for cancer genomics. We hold cancer genomic data, and this includes different types of data, molecular, biospecimen, and clinical, which we'll talk about in a little bit. The data that comes into the GDC is processed using a set of standardized pipelines. So this is many different types of data, but it's all brought in raw, processed the same way, and then stored in a flexible data model so that it can be accessed easily using a portal or an API or by any user. And then the data that we receive is submitted by sometimes academic researchers, sometimes pharmaceutical researchers, and this is processed by these pipelines and then eventually released to the research community with the GDC portal. So this is from a previous webinar, and I listed out all the data categories, but I wanted to highlight the molecular data because that is mostly what we're going to be dealing with today. We're going to talk about ways to download the molecular data and then ways to upload molecular data. So this will be light on the metadata and more about the actual files that are moving around at the GDC. So now to start with the first big section, we're going to talk about GDC data download. I put together this little flowchart just to show in general which strategy you should use. So if the size of all of your files together is less than 5 gigabytes and less than 10,000 files, you can download directly from the GDC data portal through your browser. I know that's not everyone, so anyone who either has over 5 gigabytes or over 10,000 files, we recommend using the data transfer tool in GDC API. We're going to get into later why you would choose one over another, but both of these are available for this number and amount of files and data. So the first data download tool I want to go over is the GDC data portal. Here's a screenshot of it. We're going to show it in a lot more detail in a minute or so. But basically, this is just a web-based portal. It allows for dynamic filtering of file sets by all kinds of different facets. And custom cohort selection is also supported in case you know exactly which cohort you want, or maybe you're bringing one in from a publication or something. The data is separated by open and controlled access. So everything in the GDC is either open or controlled, and all of the open access data can be accessed by anyone. You don't need to log in to do anything with open access data. If you are downloading controlled data, you will need to log in to authenticate your access and either download directly from the portal or retrieve a token. And then we have a nice little card feature to create a custom group of files that can be downloaded either from the portal or using a manifest. So now we'll get into the GDC Data Portal download demo. Here I'm using Google Chrome, and I will go over to the GDC Data Portal, which can be found at portal.gdc.cancer.gov. For files, you'll click on the repository tab here. There is additional data that is in the exploration tab, but here we're going to talk about what's in the repository. This is where the actual files are kept. So, as an example query, let's say I wanted only data from colon cancer. I only wanted data from female colon cancer patients. And to break it down a little more, we will only be selecting from the HCMI program. So this is bringing us down to only 5 patients. If we go to the File tab, we can search a little more on things that are more file specific. So let's say I was doing an RNA-seq experiment. I could click on RNA-seq. And let's say I only wanted data from the STAR pipeline, the count data. This is the number of reads for each gene that comes from the STAR pipeline. So I will click on that. And these are all open access files. There are 22 of them. And sorry, I need to break it down by gene expression quantification. The splice junction files are actually controlled access. So now we have 11 files that are the STAR counts. So I can take these and add them to my cart. And to get to the cart, you could go up to the top right and click on the cart icon. And here you can download the actual files. You would click on cart, and this would download the files directly. So they're gzips, so they're going to open automatically in my browser, but here they are. So anyways, since we're in the cart, I could show you a little bit about what's going on with these buttons. So one of the things, if you're doing a research project, that you may be aware of is that downloading the files is important, but knowing about the files is also important. Knowing things about the patients, where the DNA came from, etc. So that's where these buttons come in. The biospecimen button will download in either TSV or JSON information about the biospecimen entities that relate to the file. So this includes things like samples, portions, analytes, aliquots, slides. This will download all of the metadata about those things into either a JSON or a set of flat TSV files. Clinical button does almost the same exact thing, except it's focused more on clinical entities. So these are things like the demographic, the diagnosis, exposures. We have a list of clinical data types, but this will download all of them for the cases that are associated with your set of files. So this set of files is associated with 5 cases, which in most projects basically just means 5 patients. Sample sheet gives some very basic information in TSV format that associates the file type with the aliquot, barcode, or submitter ID, as well as the type of tumor it came from. So whether it came from a primary tumor or maybe a normal sample or a metastatic tumor, that will be listed there too. The metadata button downloads all of the metadata that we have about this file and its associated entities. So if you know you want everything, feel free to click this button. And then download, we went over that, but there's an option to download cart and manifest. And the manifest download we will get into in just a moment. So now we're going to talk about the GDC data transfer tool and how we can use that to download. So the GDC data transfer tool is a standalone application used for high performance download of GDC files. Um, this is this is available as a command line interface program as well as a UI. Today, I'm going to be demonstrating the command line interface version, and I will be assuming a little bit of knowledge, but using the command line, we are always happy to help at the GDC Help Desk. So if you have any questions about it or about this webinar, always feel free to ask us. So these files are available for Windows, Mac, and Ubuntu. If you're using a different operating system, such as Red Hat or CentOS, we recommend trying the Ubuntu version first and seeing if that works. It does work for certain versions, but it doesn't for other versions. If that doesn't work, contact the help desk, and we will be happy to help you build it from source. We have a set of directions to do that. So this transfer tool can be used to download using either plain UUIDs, so if you copy and paste UUIDs, you can download, or with manifests that come from the GDC data portal. So if you were to go to the data portal and click on manifest, that would download a manifest file. I will show you an example of that one in just a moment. So the basic command for the GDC data transfer tool, and I do want to let you know that GDC client and GDC data transfer tool are the same thing. So when I see it on the command line, I might say GDC client. I'm referring to the GDC data transfer tool. So the first command is the operation, which in this case is download. The other one is going to be upload, but we're not at that section yet. The next is to specify which files are to be downloaded, and this can be done either using a manifest -m and then the path to the manifest, or with a UUID. So a singular UUID can be pasted here. If any of the files in the manifest or the UUID are controlled access, you'll require an authentication token. So this can be specified by using the -t parameter and then the path to the token file. If there's open access, you don't have to put this parameter there. So now to demonstrate this, I have a couple of commands. So the first thing to do is I will go to the download section of my folder, and I have the GDC client stored in the folder above. So you can add this to your path. So for that, you just type GDC-client, but I didn't here. So I'm specifying the path of the GDC client. Download. And for this download demonstration, I'm only going to use open access files. And I actually put together some commands here in this text editing program, just so I don't fumble with the command line too much in front of everyone. So very simple. GDC client, download, and then some UUID. This is a UUID for an open access file. So we'll go ahead and enter, and you'll see some text coming up, and it is successfully downloaded. So if we look at the list of files here, we have this directory here with the name of the UUID that I just passed. And if we go into that directory, and we look around, you'll see that we do have this star gene counts file. So yes, this is here, this is ready. So I'll clear that, and the next thing I want to do is go to the folder above and demonstrate the GDC client using the manifest. So GDC, I'll back up. GDC client will be downloading again, -m, and then we'll put the manifest there, which is called GDC_manifest. But before I run that, I want to show you what a manifest is. So let's use this less program to show what's on the inside of one of these files. It's basically just a TSV. The first column has ID as a header, and it is just a list of UUIDs. These are unique identifiers for each file. Not to be confused with the submitter ID, which can be anything that the submitter specifies. The UUID is always in this format, and our unique one for each file. So there's some other information here, like the file name, the MD5 sum, the file size, and the state. But the most important column is the ID. If you have a list of UUIDs and you put ID as a header on the top, that will work with the data transfer tool as a manifest. It doesn't need all this other information. But it's good to keep this around for record purposes and if you need to remember what do these UUIDs correspond to. So I'm going to back out of here, and I'm going to actually run this download. So GDC client, download, and then -m we're going to use as our specification that it's going to be a manifest we will be passing. And I can tell you that everything in this manifest is open access. So I'll go ahead and run this. We don't need a token. And the files are downloading. You'll probably see that we only have 6 lines that come to 100%, but there were 16 files in that manifest. And the reason for that is that the data transfer tool actually groups these files into larger chunks for better efficiency. So if we look at our directory now, you'll see that each of the files were put into a folder with the UUID, the unique identifier, as the name. So hypothetically, if let's say some of the files happen to have the same name, they would be separated and they wouldn't overwrite each other one by one. So when moving them around, be careful if you know that some of them might have the same name. So that's the end of the demonstration for the GDC data transfer tool. And next we're going to go into the GDC download using the API. The GDC API allows users to query, submit, or download data directly from the command line. This can be accessed via the command line. We're going to be using the curl software in this webinar, but there's other ways to access it too. I know there's other softwares that can be used to access APIs, as well as Python. I know is very popular to access the API with, and there's many other ways, but those are the two that come to mind. When I talk about using the GDC API to download, I'm talking about using the GDC API to download directly. All of the GDC services, like the ones I showed you before, the data transfer tool, the GDC portal, those are both built on top of the API. So you are really using the API no matter what you do. Additionally, some third-party applications use the API since people find useful ways to parse this data out. And there are many, many endpoints, but today we are going to be focusing on the data endpoint, which is api.gdc.cancer.gov, which is the base URL for the API / endpoint, and in this case, that is /data. So here is a GDC API download command. And this This is in this example, there's a token being used, but that's not always necessary, whether open or controlled. Only the controlled will need tokens. So the first parameter, curl, the default method is the get um, command, and curl is just the software we use to access the API. The -h xauth token, and then the token variable, so not the path, but the actual token string that comes in the file, is stored in this variable here. So that's passed as a header. Then we have two parameters, remote name and remote header name. And this names the local file the same as the downloaded file. I'm going to show you why that's important during the demo. Then for the GDC API URL, that is always the same. We will be using the API data endpoint, and then the UUID of the file you would like to download, since they are unique, that will point you to the file, and that's what you put on the end. So let's go ahead and I will demonstrate that. For this, I do have a command that I will directly paste into this command line, just so I'm not fumbling around so much. And just to go through the parameters again, we have curl, the program, remote name and remote header name, so the file is saved as what it should be, what it looks like in the portal. We have the basic API URL, the data endpoint, and we will be downloading this URL, which is actually the same file we downloaded last time. So I'm going to go ahead and run this. We should get a message that it saved a file name and then this long file name. And you can see it here. So the file is now here and it has been downloaded. Now, if you don't care what the file is named, or if you want to do something special with the way that you're doing this, you can remove these parameters and it will dump the output directly to the console. It's not going to look pretty because this is a gzip file, so it's yeah, it won't work. But what you could do is use the output parameter and then name it whatever you want, and that will dump it directly into the file. And if we look at file.txt, it actually should be a gzip thing, which it is. So yeah, that is how you download using the GDC API. And one thing you might be asking is, if that only uses one file, that might not be a great use case. I'm going to show you how to do more than one of them at the end of this presentation. So let's go back to PowerPoint, and now we're done with GDC data download. We are going to get into GDC data upload. And I'm going to show you all examples of this, but GDC data upload first requires that the data is registered at the GDC. So if you have BAM files or something that need to be uploaded to the GDC, it first needs to be registered. This requires adherence to the GDC data model. There are a lot of dependencies that need to be submitted first. And then the GDC will expect a file that is a certain file size, a certain file name, and has a specific MD5 sum. And this is to verify that we are receiving the correct file and that a mistake wasn't made in perhaps uploading the wrong one. There are two data upload options. The GDC data transfer tool can be used. It's easier and can be done with a submission manifest. The one thing I do want to add is the GDC data transfer tool. I mentioned the UI version before. The UI version only works with download. So for upload, you will have to use the command line interface version. The API is more customizable and can be automated using either something like Bash scripts or Python, something like that. So another point I wanted to bring up is the difference between within a data file, it has these two properties. One is called state, and one is called file state. The state denotes whether the entity is ready for harmonization or release. It kind of tells you where the file is in its life cycle. Validated means that it was initially uploaded and hasn't been submitted for harmonization yet. Submitted means that a request for harmonization has been made for that file. So the submitter has said, this is done, we've uploaded it, it's ready, run it in the bioinformatics pipelines. And released means that the data either has been or is ready to be released to the portal. So any file you see in the portal will have the state. Any file will be in the portal in the next release will eventually have the state. Now, file state, it only applies to data files. The state actually goes across all of the different entities in the GDC, including files and just plain metadata. File state only applies to data files. So again, it has three different values. Registered means that the file has not been uploaded yet. Uploaded means that, of course, the file has been uploaded. And validated means that the MD5 sum check was verified. So when a file is uploaded to GDC, it kind of gets in this queue where a script will check the MD5 sum of the actual file against the MD5 sum that was submitted as metadata. If they match, it will go to file state validated. There's an additional file state that I didn't mention here, in that if it doesn't match, it will go to file state error. So uploading with the GDC data transfer tool is just about the same as download. You use GDC client upload to specify the operation. If you have a manifest, you do -m and then the path to the manifest. And you'll notice in this case, the manifest is in YAML format instead of a TXT or a TSV. And then you specify the token. Any upload that happens to the GDC will require a token. There's no open access upload. And then with the UUID, of course, it's just GDC client upload and then the UUID with the token path. So no surprises there. So now I can demonstrate the upload. I go to my terminal and I will change the directories to the upload folder that I created here. So when I'm going to upload, I mentioned you might need the UUID. So your question might be, how do I know what the UUID is? How do I get it? And I'll show you a little bit of what happened, what I did to prepare for this. So first, the uploaded metadata, I can show it here. This JSON file was uploaded not too long ago. They are for submitted online reads. I uploaded 5 of them, and the data for each of these is specified. So for example, this one right here, which is the one we'll be using, it is a submitted online reads file. It was associated with this read group here. This is for a test project, so we don't worry too much about the data quality. The file name is expected to be file2.bam. The MD5 sum is expected to be this. The file size is 16 bytes. And then there's some additional information that just kind of gives some categories and more information about what the file is. So in this case, it would be whole exome sequencing. And if it's paired end, it would have a repaired number of R1. You could also put R2 there, but So let's look at this file. So this was uploaded to the submission portal, and that makes the file registered. Um, One more thing I do want to point out is you'll notice there's no actual UUID here. There was none specified, and that is because they are they are assigned at creation. So I uploaded these. I actually used the API to upload them, and what came out of this was this file, a JSON file that kind of shows how the transaction went. And um, here is what we get. Let's find that second file, which should be right about here. So we have file internal 2, that's the submitter ID. The ID is right here. So that's the one that I'll take. There are other ways that this can be retrieved. For example, if you didn't save the output, or if you just have way too much, and I know going through and copying, pasting the ID is not a reasonable thing to expect someone to do for like 1,000 files. So, I just stuck there. So yeah, there are other ways to do it, and if you have any questions, feel free to contact the GDC help desk. So what we'll do is GDC client upload that UUID and -t token.txt that has the token string in it. So before I do this, I want to let you know that this UUID corresponds to file2.bam. Now, the GDC client is a convenient program in that it will figure out using the API what the file name is supposed to be, and then pull that file name from the directory that you're in. So in this case, it should find file2.bam. I will not have to specify, but if you need to, you also can. So there, it went ahead and uploaded. So now we can actually look at this in the submission portal. So I will go to the submission portal. That is the GraphQL. I'll go ahead and sign in to here. I've access. Usually there'd be a list of projects here, but I have access to a lot of them, so I'll just go directly to GDC internal dashboard. And if you remember, this was a submitted unaligned read. So if you go to browse and you check the data type, you can click on submitted unaligned read, which is right here. So if you remember, this was BAM file internal 2. This is right here. And you can see that the state is validated and the file state is uploaded. Now, let's say you uploaded the file and you realize that's the wrong file and you need to delete it. Very easy. You just do -- delete using the same command, and that should finish it off. Sometimes it takes a second. Oh, there is an internal server error. So that usually will just delete the file. So going back to the oh, one more thing I wanted to show is the manifest. So if you click on manifest here, this will download a manifest for the file that you would like to upload. So I actually have one ready to go here. It's called manifest bam file internal 2.yaml, since that's the name. And I'll just run the same command I did before, except I have -m manifest and then the token. This should do the same thing. This would have worked had it been deleted. I can try again. real quick. There we go. So the file is deleted, and now I can actually upload it. Apologies for the previous bug. There we go. So now that the file is deleted, I can upload it again, and it works using the YAML file. So now I will go back to the PowerPoint, and uploading with the GDC API, here's the basic command here. The main difference you'll notice is that you don't, in this case, you will have to specify exactly which file is being uploaded. So while the GDC data transfer tool, GDC client, can auto-detect, this will not. So if I was uploading a file called file.fastq.gz, I would need to specify that. But most of this is the same. In this case, we're going to use the xput method. In this case, we will need a token in the header. I just talked about how we have to specify the data. And then we will use the regular API URL, the submission endpoint, which we will put the program and project in. So if your program was something like TCGA-BRCA, TCGA would go here and BRCA would go here. And then /files is the endpoint. And then at the end of files, you would put the UUID that corresponds to the file that you are uploading. So in our case, I'm going to real quick, since we're using that same one, delete this one. And while it's deleting, I will grab this command here. Okay, so here we have the command. We have curl. We use the method x and then put put. The header is xauth token, and then token is the this is the token string stored as a variable named token. Then for the data parameter, we will put at file2.bam. The name of the file is file2.bam, but the at has to be there to let curl know that we want to submit the data that's inside the file rather than just the text file2.bam as a string. So that is important. The URL is the regular gdc.cancer.gov URL. The program, the project, in this case GDC internal, it's a test project that we use, files, and then the UUID that I specified here. So I'm going to go ahead, oh, whoops. So that is a problem that can sometimes happen. The dashes need to be dashes, and they can't be those fancy long ones that come from word processors. Whoops. Sorry, I forgot one thing, and that was submission. That was required. And so the upload happens without doing anything. If you need to see something, you need to specify the output path and just do like file.test or something. And then you will see an actual progress bar. So that is upload with the API. And I will go to the frequently asked questions. So one question that we get a lot is, how can I download and upload manifest? The button only serves an empty file. So this is a known issue that's in the GDC data submission portal. If you try to download an upload manifest for hundreds or thousands of files, it will time out and serve an empty file. So my team manages the GDC help desk, and we help users with this. And if you come to us with this question, we're happy to help you generate a manifest. But here, I would like to show you how to actually do this. So in this case, I have a list of UUIDs to upload. So there's 5 UUIDs, and I want to upload them all. So, and I want to generate a manifest for this. So I'll start a for loop in Bash for x in, and then I will add that as a variable, do, and then I actually have this command ready to go. So we have curl, pass the token, and then we will be going to the, can't believe that's submissions. It should be submission, GDC internal, then the manifest endpoint, that's where you retrieve a manifest, and we're going to specify the ID by passing this IDs parameter equals, and then each of those UUIDs. So we're going to run them in sequence, and we're going to concatenate them to a file called bigmanifest.yaml. And then we'll put done here, and this should run. Oh, sorry, that's dash in the H. Got me. Okay, 1, 2, 3, 4, 5. So they all ran. And if we look at, oh, not giant. I named this one big manifest. It should have all of the manifests in sequence here. But the problem is with GDC manifests, it just needs to have the files header at the top, and these bottom ones are not supposed to be there. So it's just a small hack that I do is I will grep out the file, the files header. I will put that into a temporary file, move the temporary file to the manifest. So I have big manifest here, and then I will just add it manually to the top. So this is a valid GDC manifest. Now, I only did this for 5 files for the purposes of time, but if you're uploading like 1,000 files, this is an easy way to get this going. And like I said, this is something that we are happy to help you do at the GDC help desk. The second question is, how can I download multiple files via the GDC API? And I'm running out of time, so I'm not going to go through this one, but it's basically the same thing I showed you before, but replace the UUID in the API with the variable that you're looping through. So yeah, you can also do this with Python if you have a Python script that's connecting to the GDC API. A really quick for loop should take care of that. And the third frequently asked question is, can GDC data be downloaded directly from cloud services? And the short answer is no. We don't have GDC data available to be downloaded directly from clouds at the GDC. However, there is another group called the Cancer Research Data Commons. We work with them closely, and they have the same data as the GDC. And I'm not affiliated with them, so I can't give you too many details, but I know that they do have options and ways to download directly from the cloud. So feel free to reach out to the Cancer Research Data Commons if you have any questions about that. With that, here are some GDC links to the portal, documentation, and the website. As always, I'd like to mention that my team manages the GDC help desk. So if you have any questions that weren't answered here or just about anything with the GDC, feel free to contact us at this address down here. You can also find it on the gdc.cancer.gov website under the support tab. That is it, and now we will take any questions.
Thanks, Bill. Hi, everyone. Please place your questions in the Q&A. We did have a question about the recording of this video, and it will be made available on the GDC website that Bill just showed once we complete processing the video. So we will have this recording available. Does anybody have any questions? Just give it a minute or so. I think, Bill, you may have covered a lot of the questions in the FAQ overview.
Oh, good to hear.
All right, if nobody has any questions, on behalf of the GDC team, I'd like to wish everybody a happy and safe holiday. There's actually one question did come up. Thanks for the kind explanations. What about FASTQ protected files? How should we download those files?
Yeah, that's so fast queues aren't available directly at the GDC. If you look at the portal, which I'll go back to, and you look at the data types, we don't actually have any fast queue here. And that's because we store all of our molecular data as BAM files. So if you were to download the BAM files, these are all controlled, but we do keep all of the unaligned reads so that we don't throw anything out. So you could recreate the original FASTQ file by downloading these. And we do tag them by read group. So if it was generated using multiple FASTQ files, you will be able to separate them out into multiple FASTQ files.
Okay. All right, are there any other questions? Okay, if not, then you'll all have a happy holiday. Take care, everybody.
Thank you, everyone.